It is a great pleasure to have today with us in our Bar-Ilan University Vision Science Seminar Professor Kendrick Kay from the University of Minnesota, Twin Cities. Professor Kay did his undergraduate studies in philosophy at Harvard and then his PhD in psychology at UC Berkeley under the supervision of Professor Jack Gallant. He continued to a postdoc at Stanford working with Professor Brian Wendell. After being a visiting scholar in the psychology department at the Washington University in St. Louis, he joined the University of Minnesota Twin Cities as faculty, where is part of the Center for Magnetic Resonance Research in the Department of Radiology and also affiliated with the Department of Psychology. He has published many research articles and multiple reviews, commentaries, and book chapters. Many of his publications are in top leading journals, including in Nature, Nature Neuroscience, Nature Communications, Nature Machine Intelligence, Current Biology, Trends in Cognitive Science, and many more. He has an H index of 48 according to Google Scholar. He has given dozens of invited talks across the world and has held and is still holding multiple editorial roles. His research is consistently funded by multiple funding agencies. Professor Kay is the head of the Computational Visual Neuroscience Lab that focuses on understanding the computational principles by which the brain processes visual information, and his research lies at the intersection of cognitive neuroscience, fMRI methods, and computational modeling. He's known for advancing high-resolution fMRI methods and for developing quantitative models that describe the neural computations in the visual cortex. Professor Kay's lab has created large-scale datasets and tools that have become widely used across the human neuroscience community. So I'd like to welcome you, Kendrick, to our seminar to hear about your exciting research, and thanks so much for joining us, and especially at this very early hour.
Thanks, Sharon, for that. Very gracious. Hi, everyone. Thanks for showing up. I think I'm awake. I can probably do this. Sharon, please interrupt me if I'm going over, or if there's a question or something's messed up. Obviously, let me know. I'll keep this to about an hour, right? And please, I'll stop at various points for questions. But I kind of want to cover a variety of things, so I will go kind of fast. And I know that's a little tricky. And there's probably a breadth of different expertise here, but yeah, I'll stop periodically for some questions throughout. Now, I guess this is a vision science seminar, so maybe it's strange that I have the word cognitive in here. I will say that my research, I guess I've been drifting away from vision a little bit, and maybe you'll see that in this talk. But I think in a good way, because I think I deliberately try to structure my lab's research to try to be broad. And certainly this is not all of neuroscience or psychology, but I do my best to try to touch different fields and try to bring different things together. And I hope you'll see some of that in this talk. The talk today is very data-y, so we maybe won't hear a lot about like vision, kind of the theoretical framework of vision. I'll try to work in a little bit, but it's really about data. And the reason is I think data is really important for what it is that at least I try to do. So quickly an intro to kind of how I think about things, and then kind of three mini talks, and I'll kind of stop after each one because they're all a bit different. And I want to at least touch upon each of these things with some depth, just to give you a sense. This is sort of an introductory introduction to how I think about vision and the brain and computation. Okay, so first, some very gentle intro. I guess fundamentally, I would call myself a neuroscientist, a visual, computational, maybe cognitive neuroscientist. I mean, these labels are a little fuzzy. And okay, from that point of view, I guess I don't start with behavior. And I know many people, and it's totally valid to kind of focus on behavior and like behavioral performance and perception and things like that, in which I know floats many people's boats. But as a neuroscientist, I'm going to start with the brain. Okay, so the brain is obviously an extremely complicated organ, and exists at multiple scales and temporal scales, and kind of attuned to the measurement technique, which I primarily use as fMRI. We're going to use this phrase neural population, so we can measure hundreds of thousands of neurons in a voxel, and so without making too strong of a claim about which spatial scale is most relevant, let's just try to model and measure things at the finest scale that we can access using the techniques we have. And we want to do this in a quantitative/computational sense. Okay, we want to build serious models of how brains work. Now, why is this hard when it comes to vision and presumably any other cognitive faculty done by the brain? I would say that the main challenge is getting the right data, enough data, the right data, high quality data, so measurement. And in vision, it often boils down to the space of possible visual stimuli, and it's vast, obviously, and there's many different types and many different dimensions to probe. I don't want to leave out cognition, and you'll see a little bit of that later in this talk, but let's just think about visual space. We need to sample the space, and have many questions about how to do that well. And the challenge here is getting good data is expensive, both time, resources, your time, and so forth. Okay, so that kind of motivates the next two sections of this talk. Like, we need better data, according to this view. Now, one choice people face when getting expensive data is how to do that. So there's a trade-off between the number of distinct humans, if you're doing human neuroscience, or organisms more generally, that you sample from, and the kind of extent of the experimental manipulations that you sample. We made this figure a couple years ago now, just plotting something very simple. So each dot is a publicly available fMRI data set out there. So a little biased here to human neuroimaging. And we're just plotting x-axis of just sheer number of hours of data recorded using fMRI from a person. And the y-axis is the number of distinct persons sampled in the data set. And notice it's on a log axis. So you can see there's a huge variety of different points in this space that there are existing data sets sample. So up here, there's tens of thousands of subjects possibly, and very little amount of data per person, versus the lower right, where the priority is on precise or dense sampling per person. So many hours of data, but only a small number of subjects. So NSD is it's a subject of the next little section of the talk, and we deliberately chose NSD to kind of live in the lower right, reflecting our preference for data sets that get precise estimates for a small number of subjects. We wrote kind of a perspective piece, kind of outlining how we think about this, which many people subscribe to, but we're not saying it's the only way to collect data. There are, of course, advantages to collecting data way out here. So just briefly, our stance, which probably many of you agree with, coming from a vision background. So why do we want to dedicate most of our data collection to a small set of individuals? Well, fundamentally, we're trying to characterize each individual subject. Every individual is unique, both anatomically, functionally, behaviorally. So it makes a lot of sense that's an object of, or that's the target of our research. One thing we can avoid, and that's typical in neuroimaging, is kind of, putting all the brains into some standard space and averaging. You lose a lot of information. You possibly introduce artifacts. So like, if we can avoid that to the extent possible, that seems like a major advantage. The general goal is to show establish evidence for some theory or find some particular phenomenon and show it in every individual subject. That's kind of the intention here, if we can do it. And ultimately, if we take a modeling approach, if you have some sort of model that incorporates various principles about how visual systems work, for example, we presume that, what we're going to learn, if we see it in a couple of people, 2, 3, 4, 5, 6, 7, 8 people, like the principles we learn are probably going to generalize. And we do have evidence of this in what the field has learned about the human visual system thus far. Okay. One last thing, kind of introductory, is this idea, and this is basically how we pitched NSD, is like, this is kind of a different way of doing neuroimaging. And for lack of a better term, we call it intensive neuroimaging, and we kind of sketch out a vision of this type of data set. And there are more and more of these data sets coming online in the last year or two. And so who knows what the future is going to see. But the idea is to collect a lot or a massive amount of data. So like more than typical, more than most people would want to do. And we're actually currently doing a new data set, which you'll hear briefly about also, in a small number of people. And the goal is to not explore any one particular hypothesis, but to design a kind of rich data set that we know would be very intriguing from many different points of view, to answer many different hypotheses and be reused for many different uses. And biased by what we're trying to do, kind of the type of data that was good for modeling. So sampling things very precisely, choosing stimuli if we can well, and kind of building a data set that can drive kind of machine learning approaches and computational modeling approaches. It's useful to think what it's not, and this is not, again, we're not saying this is the only way to do neuroimaging or neuroscience measurements. One thing we try to emphasize when designing these data sets is basically that word design. So it actually requires, or ideally requires, a lot of thought about what type of data to get. It's not just taking off-the-shelf experiments and just increasing the number of trials and sessions. That's probably not as useful as actually spending some time to craft a well-designed experiment. In neuroimaging, there's a lot of resting state, so it definitely has value, but it's not, and a lot of people collect a ton of resting state, and that's useful for some questions, but it's not really what we're talking about here. Okay, so that's super brief. I don't want to spend too long on these kind of high-level generalities, but let me stop there to take a pause and see if there's any comments or questions on that. Don't be shy.
Kendrick, yeah, I'll
Yeah.
just ask you a quick question. So, the individuals, how do you choose them exactly? Is it going to be just naive individuals in general? Do you recommend that, or people from labs?
Yeah, great question. I will touch upon this in section 3, but just briefly, these are highly biased samples. In vision or maybe visual neuroimaging, it's common for labs to like scan themselves, but for these data sets you're going to hear about, we definitely could not do that, as you say, because we needed naive subjects, so we couldn't just scan ourselves. But I don't think there's anything necessarily wrong with doing that if the experiment that you design, is compatible with that. But we did expend a lot of effort on subject selection in number 3 here, which I will get to. But yeah, these are not like choose randomly, and for a good reason, as you might suspect. I mean, these are not easy experiments to be a subject in.
Yeah, of
Any
course. Thanks.
any other questions? Okay, so as I kind of foreshadowed, NSD is an example of one of these massive or somewhat crazy data sets. This is all kind of done already, and the data's out there. A lot of people are using it. But so I just want to kind of review a little bit historically what it is in case you haven't heard about it. At the time, we decided, or we knew that, I guess the field wanted this data set, or we actually sent out a survey kind of on Twitter back when Twitter was Twitter, just getting a sense of people out there and would people want this? Do people care? And so we did some informal surveying. So as I mentioned, you can think about in neural measurement, like in vision specifically, it's about sampling a lot of stimuli to drive like understanding the various dimensions that are represented in various visual areas, or at least that's a common way to think about the visual system. So Thomas Nasleris and we were kind of the co-leads on this. We kind of thought of this idea that the field needed this data set, and Emily Allen was a postdoc I was able to hire to help carry out the data acquisition effort. And basically we designed this data set, collected it at high field fMRI. So that's at the MRI center here. We have several 7Ts, and we have a 10.5T, which can be used for human functional MRI in general. And yeah, the data set is really deep and extensive and well documented, I would hope. And it's all laid out in this paper. Let's just jump to the experiment. And in some sense, the experiment is super straightforward. And this was deliberate. In designing this data set, we didn't want to innovate or make things too crazy. So the experiment itself, in terms of the stimuli and how it's presented, was deliberately kept fairly straightforward, just so it wouldn't break. There's many other things that can break with fMRI, let me put it that way. So in this experiment, you essentially fixate in the whole time, which is hard, of course, and there are, of course, microscots potentially. You fixate and you are exposed to these complex scenes, color natural scenes taken from a computer vision database called Microsoft Common Objects in Context, or COCO. These are square cropped, 8.4 degrees on a side, presented for 3 seconds, which is an eternity, obviously, for vision, but it's done like that because fMRI is a very slow signal. So we kept the image up for 3 seconds, and there's essentially a 1-second cap. So every image you get, you're making a new old judgment. So we have a couple of repeats of each image. These are distributed out across scan sessions, across weeks, across months. So this is a memory task in a sense, and you're judging, have you seen this particular image thus far in the experiment? So we have some behavior for every trial. That's good to keep tabs on the subjects, of course, and central fixation and complex visual stimulation. So this is not the experiment. This is just to give you a sense. Hopefully this comes pretty well over Zoom just to show you the types of images that come in this COCO database. So teddy bears, people, action scenes, colorful scenes, animals, objects, indoor environments, outdoor environments, so on and so forth. Now that the data has actually been released, there's been some commentary that, this is not the only sampling of visual stimuli that one could do, and there definitely are statistical biases in this data set that people have commented on. But at the time, we thought this was a good place to start. Okay, some of this next couple slides is kind of very specific to MRI, so probably not all of you care or do it. So I'll go kind of quickly. We did whole brain 70 fMRI at a pretty high spatial resolution. In terms of how much data, we had 8 humans who stuck through it. Each session was an hour of actual data, and they came for 30 to 40 scan sessions each. If you add up all the numbers, across all subjects, there's 213,000 trials, where a trial is, one presentation of one of these images with a button press. So it's big, but certainly it's possible to get bigger. And so there's choices we have to make about how long we're willing to do this for. A lot of fMRI data quality issues. This is part of what my lab does. So we spend a lot of time crafting, customizing, and innovating some methods for making the best possible data, at least from our point of view. And I will just briefly flash a slide about what that's all about. I know that's not what everyone does. So this is that slide. fMRI preprocessing is a big nightmare for anyone who does it, but basically space and time issues. So let me just show you a movie, since a movie is intuitive. This is showing, after basic fMRI preprocessing, motion correction, registration, undistortion due to field inhomogeneities. This shows you 1 frame per session. So this is the brain of this one subject coming back, a week later, typically, and getting scanned for, a whole hour separated into runs. And we're looking for spatial stability here. You can see it's already highly detailed, so that's good to look at your data in terms of resolution and image artifacts. But we also see, hopefully, again, it comes through over Zoom. I guess I'll go slow, that it's pretty stable. One thing we are not looking at is, how anatomically correct it is. We would have to look at anatomical images for that. But simple visual inspections, I think, are super critical. In fact, lately, I've been thinking more about this and actually looking at other data sets out there and thinking that people should more routinely make these types of visualizations. So we spend a while optimizing those things. There's also taking the time series data and extracting the relevant bold response from it. So just an overview of that process. The goal was to estimate kind of a brain map or entire beta weights in a sense. So every voxel, what is the response observed at that voxel to every single trial in the data set? We cobbled together some statistical ideas of how to do that optimally, at least from our point of view. That involves estimating different hemodynamic response function, which is this kind of temporal response function that you can estimate if done well. We do that per voxel. We also do some nuisance regression based on looking at what the data is like in voxels not of interest. And finally, using some ridge regression to stabilize the single trial estimates. Basically, from an fMRI point of view, the experiment, even though it's slow from a visual point of view, it's actually pretty fast for fMRI. And so there's some collinearity issues with the response from subsequent trials. So ridge regression is a very simple and established technique to possibly improve the estimates for individual trials. Anyway, so we cover this in the paper, and these methods are general, and we made a standalone little toolbox that can be applied to other fMRI data sets. All right, so after all is said and done, we get this large data set, 8 subjects looking at thousands of images. In the paper, we just do a teaser of look, there seems to be good data here. And this is more of a high level look at the data than signal quality metrics. Here, we're just showing a TS&E plot. So it's a kind of a visualization technique of condensing high dimensional data onto a screen. Showing and essentially what's happening, it's positioning these individual stimuli according to their brain-wise similarity. So pick a brain region, measure the neural similarity between the representations of two images, and try to optimize the position of these stimuli according to that. Here in this panel C, we're looking at anterior ventral temporal cortex, so well, deliberately very large region. It's actually very hard to get good data out here. But anyway, if you pull the voxel signals from and basically high-level ventral stream stuff and use the data to drive the positioning of the stimuli, you get seemingly sensible results. Like in this zoomed-in region, you get these round food like edible donuts and apples and such. And of course, you could repeat this for different parts of the brain, and we see some patterns that we sort of expect based on prior work. So that's kind of just a teaser of like the richness and the vast scale of the database now that we've generated. And one other teaser we put in the paper was this very computational neuroscience idea, which is to use the vast scale of the data set to drive neural network training. So instead of training a neural network on some computer vision objective, which is totally fine, we can just directly fit models and make them reproduce the brain's essentially stimulus response mapping. A lot of details I don't want to go into, but the main result is it works. So the x-axis here is just the sheer amount of data you can give to the training of a neural network from 1,000 trials to up to 27,000 trials. And these so-called brain optimized networks are in red and orange here, and you can measure the out-of-sample cross-validation performance on the y-axis. And basically performance starts pretty poor compared to some baseline models you see here, but eventually you have enough training data, these models are able to get trained well enough so that the performance starts to be seemingly satisfactory. There's more about this type of approach in this standalone paper here. Okay, so we released this data set to the visual neuroscience community. There's many reuses of the data. Just one that has become prominent is very cognitive. There's kind of a discovery or maybe rediscovery, depending on your read of the literature, of selectivity in the ventral stream for food. We all know that there are these sort of dominant types of categories that seem to be in the ventral stream, but food was a little controversial, and basically 3 independent groups, and we were involved in one of them, kind of rediscovered kind of converging analyses, of course the same data set, but with different analysis approaches for sure, and there's definitely differences across these 3 papers, but long story short, there's kind of this nice example of science, like almost collaboration in a sense, all looking at the same data set and coming to similar conclusions. As I said, yeah, this has been widely used, maybe not as widely used as other kind of more general purpose neuroimaging data sets out there, but I think it was successful from that point of view of creating a resource that actually drives many different research questions. So let me pause there. I know that was quick, and see if there's any questions about NSD.
Kendrick, just to clarify, so the task was on every trial to press, say, one button if you saw the image before, or another button if you didn't see it.
That's right. Yeah, the choice of task was a hard one. Obviously, there's
Yes.
many different ways we could have done it. We were trying to balance several criteria. For example, we wanted something for sure for subjects to do, otherwise they're going to fall asleep. Everyone knows who does fMRI. We wanted something that might actually be interesting in and of itself. So some memory researchers have used the behavioral data alone or in conjunction with the fMRI data to say something about how memory works, this very long-term memory aspect. We wanted also a task that was kind of general purpose as far as we understood about you don't want to necessarily bias the visual system to focus on any one particular thing. So that was another consideration.
Okay, and another quick question I have is, I imagine they were all presented in upright configuration. You didn't have a separate experiment or the same one in which you were flipping them upside down, no?
Actually, it's funny you bring that up. There actually is. So we did roughly
Hmm.
40 sessions of
Okay.
this, which is a lot, but there was an extra 2 experiments we tacked on to these same subjects. It's called NSD synthetic and NSD imagery. So NSD imagery is a whole
Hmm.
scan session trying to probe mental imagery, which is one of the main topics that my collaborator Thomas cares about. The NSD synthetic is a bunch of kind of, gratings and noise patterns, and one of the manipulations I wanted to put in was upside down scenes. So NSD synthetic, I mean, it's just one scan session, but some data is better than no data. So yeah, if
Yeah.
there's something of interest regarding that manipulation, it actually exists for these eight people.
Okay. Thank you.
Any other questions about NSD?
I just wanted to ask about eye movements, whether you really know that they actually fixated, because when I looked at the images, it was like my eyes were completely drawn to other
Yeah.
places in the image and not the fixation point. So I wonder if you did control for that. I mean, even if you didn't, from my point of view, that's okay because, it's been shown that people are consistently looking at the same places anyway. So likely that would repeat across sessions, but I'm just wondering if you.
Yeah. No, good question. In short, eye tracking is the bane of my current existence. So for our current data set, so for NSD, we did some eye tracking during the scanning. It hit or miss. Success rate maybe half the sessions, maybe. It's very hard due to annoying setup reasons. Um, so what I can say is we have reasonable evidence in a couple sessions that these humans, obviously we didn't get good data for all 40 sessions, but we have some data, it's in supplementary figure 5 or something, that shows, as far as I can tell, my best guess as to how well these people fixate in this type of experiment. It looks pretty good. There's maybe one subject that we noticed clear small saccades, like they couldn't
Mhm.
help but like kind of move their eyes a degree or two, maybe that way or that way. But most subjects on the whole seem pretty good, or as much as you can actually fixate for long periods of time on a dot.
Yeah.
Um, there are some
Okay.
analyses. I mean, you can derive retinotopic organization from analyzing the responses to this, so that implies that it's at least stable enough to, do that type of
Yeah.
thing. And
Yeah, definitely.
we show that in the paper. But yeah, in our current data set, which is really the next topic, we are trying to make it eye tracking in the scanner work, but it continues to be a headache.
Yeah. Yeah. It's not as easy in the lab. Okay. Thank you. Thank you.
Okay, thanks. Okay, let me move on. So VCD, in some funny sense, is completely different. It's still vision, it's still fMRI, so there's definitely some similarities, but and it's something that I think is really important I mean, switching gears a little bit from a more theoretical perspective. We just heard about NSD, and it's essentially just imagining that largely we just need to sample stimuli, but most of you hopefully agree, like that is definitely not the full picture of how humans see. And specifically this issue of what do you have people do, like overtly or covertly or explicitly or implicitly during the experiment. So tasks, like what did you instruct them to engage in during the experiment? In NSD, as we briefly discussed, it was this kind of new old or memory recognition task, but that was not really, that's just one possible task. Let me put it that way. So somewhat recently, I wrote a review with some colleagues on this idea, kind of probing people, how different people think about this. I guess our target was visual neuroscience and maybe not vision broadly speaking, including like psychophysics, and just taking a kind of neuroscience point of view and basically pointing out that visual neuroscientists often, I mean, I'm not saying it's never been studied, but by and large, the default is just to think of, probing the visual system as like a stimulus response relationship. Briefly, we can define a task as and there are different definitions, of course, one can propose, but the goal and strategies used by this particular observer, this particular subject or participant, in the context that you engage visual stimuli with them. Why do we care? Well, I think we all agree that it's interesting to think about how brains accomplish tasks. And of course, with the rise of AI and the ability for AI to do all sorts of these powerful tasks, I will say the word task has a different meaning in AI and machine learning. But tasks are important and interesting in terms of how humans accomplish them. Even more importantly, it matters to NSD, for example. So NSD, if you think of as like measuring the representation of various stimuli in the brain, well, depending on the task, that can actually change how those measurements turn out. So the representations, if you will change possibly depending on what task the subject is engaged in. And when you stop to think about it, and again, it's kind of in this article, it's actually a pretty tricky to think about what are tasks and how do we know what the tasks are and how do we individuate tasks and how do we even talk about tasks. So it's a I think interesting topic. Maybe I'll skip this slide. This is just one of the figures in the paper where it's really just me this is definitely not like a quantitative slide. It's just a schematic of how I think about different possible tasks that we might use in a psychology or neuroscience experiment. So I encourage you to check out the review if you want to know more about how I think about this figure. Anyway, so that motivates this next data set that we're currently collecting, and it's a very different beast than NSD. This is a collaborative project with Clay Curtis at NYU, and Elena is a postdoc, but now faculty at York University, who basically contributed a lot to the design and implementation of this somewhat crazy experiment, which I'll show you in a second. Okay, so the overall goal of this data set, kind of foreshadowed by what I just said on the previous slide, was to sample deliberately many different tasks. So not just, for example, in NSD, this kind of memory recognition task, but a variety of different tasks and try to understand and just, well, first just observe what happens under different task contexts on visually driven activity. That's the primary goal. And so similar to NSD, we're doing this with an eye towards making this rich data set that hopefully many people will find interesting. It's public, freely available, so on and so forth. Now, one of the issues is there are obviously infinitely many tasks and infinitely many stimuli, so just getting a sizable, reasonable data set is going to be a very large data set. So many, many scan sessions, and we're up to essentially 15 sessions already. I think we probably will get at least 15 more sessions, so it's a lot of data. To constrain this experiment, which is very ill-defined, we're going to first have this idea that at least the stimuli should be identical across different task contexts. And so a lot of work was in the design to make that possible. Like NSD, it's going to be high quality data. We're going to do all our fancy MRI tricks and statistical stuff, blah, blah, blah. And I think, of course, there's some precursors of this out there, but I think at the scale and diversity that we're shooting for, I don't think there's really anything like this out there. In contrast to NSD, now there's many kind of large image-oriented or video-oriented visual data sets kind of out there in that mindset, but this type of data set I don't think has really been done. I'd be happy to hear if I'm missing something out there. And so I think it's a bit or quite risky because like I'm not sure we're ready for this data set. Hopefully you'll see what I mean in a couple slides. Okay, so general design principles. We opted for nice classic vision science stimuli. So control the stimuli. You'll see some of this in a second. So Gabors and random dot kinetograms and so forth. And we want to choose carefully because, again, we want to sample well and we don't have infinite time. So we're going to design these stimuli and tasks. We want to try to sample task space widely, subject to the constraint that there's only so many scan sessions we can get. We also want to sample stimulus space widely because, we don't want to just use a single type of stimulus because we might be missing, to the extent that a visual system is specialized for different visual attributes, we don't want to be missing part of the picture here. So we kind of want to do two things simultaneously, and making it harder, we want to cross stimulus types and tasks. So essentially, if possible, do all the tasks with all of the stimuli. So that makes it a combinatorial explosion already. And in terms of choosing what tasks, we kind of surveyed, actually more than kind of, Clay surveyed VSS and kind of looked at what does the field like to do? Like what progress have we made? What type of tasks have people chosen for various reasons? And we're kind of using that as a guidepost here to constructing the experiment. So let's look at the experiment. This is a bird's eye view. There's a million details that I cannot have time to explain. But essentially, we have 5 different stimulus types here. So oriented Gabors, a dot whose position is varied around the visual field, random dot motion, briefly presented with some direction of motion and some coherence level of a couple different types of objects, and then a couple selected natural scenes. So we have these very different types of stimuli probing different aspects of vision. And we have on the columns 10 tasks, essentially. And I say this with some hesitation because it's really complicated to even know how to describe a task and to consider whether it's actually, or even what to name it. So these are just words, right? And by no means are you meant to understand what the task is from the words alone. But briefly, fixation. So there's a fixation task where you're ignoring the stimuli, doing some demanding thing at the fixation. This contrast task, you're essentially looking for a brief, somewhat subtle contrast change in the stimulus. What we're calling categorization is kind of a, is it a Gabor or dot or motion type of judgment. Feature categorization is about the property that's relevant mostly, right? So the orientation or the position or the motion direction. There's a working memory task where you're encouraging subjects to keep a briefly presented stimulus, the attribute in mind, and keep it over a delay, so very classic manipulation. There's also a long-term memory context where the subjects learn associations between these stimuli and have to recall the paired associate when cued with one of them. There's also a visual imagery one where instead of delivering the visual stimulus, the subject is asked or rewarded to internally generate that stimulus using mental imagery. And there's also these last three, which are more naturalistic. This is essentially what type of object is a dominant object. It's intended for the natural scenes, as you see out here. This one is like where in the visual field is it, left, right, or center. So it's kind of a where task, whereas this is a what task. And this last one is more of a how task. So essentially, how do you feel about interacting with this object, if at all? Okay, so the colors here represent the cells that we actually sampled, the crossings that we actually tried to get a ton of trials within. Some of the cells don't really make sense, and I don't want to get into the details there. I don't have time. The purples you can think of as the more classic VSS style tasks, whereas the gold are a bit more strange because they're naturalistic and get into tasks that I don't think we have a lot of prior literature on. That's the bird's eye view of this experiment. And now I'll just show you, oops, one important thing, sorry about that, is given this crossing, which is not fully crossed, so we're not sampling these squares because it's a little strange. But basically we can ask two types of questions at least. Like one is for a given type of task, or at least how we think about it, we can look at how the brain responses are across very different types of stimuli. And then we can also go in the horizontal direction, which is for the same retinally delivered stimulus, except imagery is the exception. There is no retinal stimulus. We can look at brain responses in the visual system and beyond across different task contexts. Okay, so let me show you the actual experiment, or at least a sample of trials within the experiment, and then I'll stop for questions. I am assuming there probably are some. Let me jump to the middle. So we set it up as essentially a blocked design, where in the sense of like you do multiple trials of the same type within a block. So let me just find a block. Okay, here we go. So in this block, the subjects have 4 seconds to remember what this task is. So it's actually pretty challenging. There's upwards of 30 different task instruction screens with their own button mappings. So there was a whole training procedure for the subjects in order for them to prepare for them for this. So you get 4 seconds to remind yourself what this task is. So you're going to be seeing these single dot stimuli, and you're going to be making, this is the working memory task basically, where you're actually just holding the position of the dot in working memory and then judging is the test dot counterclockwise or clockwise relative to the first dot. So here we go. So you cue left. There are 2 stimulus locations basically, one on the left, one on the right. So we just got cued left. We saw a dot. You should be holding central fixation. You're in the delay period. Oops, and that's the test dot. So you're making a judgment when the second stimulus comes up. Let's do another one. All right, here we go. You're going to get a spatial cue right. Okay, now you're holding this dot in working memory as you fixate and ignore these little luminance changes. That's not relevant for this current task. There's a delay period. Oh, there you go. And then you get the test stimulus and you make your button press. So just a series of trials, kind of keep it simple, kind of classic trial-oriented cognitive neuroscience. And this is done for all of the different tasks. So here's the next one. You're judging the rotation of the object that you're going to be presented, and you have a two-way judgment. Is the object more forwards looking, like towards you, or is it more sideways moving? So rotation of the object was one of the dimensions of the objects that we chose to manipulate in order to support this task. So it's a two-way judgment. Here we go. You get your spatial cue left. You try to think about what that object was, and you make your judgment. Next trial, cue left. You get your object. The objects are on for 1 second. Keep it brief. Here's another object. You're making this kind of heading or rotation judgment. Here's a cue right one. You make a judgment there. So notice, as you see, there is this stimulus on the other side, which serves as a nice control comparison case. Will generate responses, of course, in the brain. And anyway, so there's a left-right cue. It goes on and on for, in this case, 8 trials. So hopefully that gives you a sense of the structure of the experiment. There's also natural scenes, which are not left-right. So maybe I'll just show you one example of that. So let me get to the start. Okay, so you get a 4-second task instruction screen. Fairly simple. Like the goal here wasn't necessarily to tax the subject into doing something, really crazy behaviorally. It's an indoor-outdoor judgment. Okay, I'm ready. I get my cue, which is non-informative. It's a central presentation. I get my image. I make a response. Okay, next image, make a response. So there's a lot of design involved in this experiment. Choosing, whoops, I just accidentally, sorry about that. Let me reshare my screen. Okay, a lot of design choices. Let me know if,
No,
oh shoot.
sorry,
Oh,
it's not shared
yeah.
yet.
Okay, got it.
Yeah, it is.
Great. So hopefully that gives you a feel of what the experiment's like. A lot of design choices. We need to write it all out and motivate all the little choices, but it's a work in progress. We're collecting data still. I just want to show you just this slide, just to give you some flavor of what we did. So we stuck 20 from a phone recruitment. We chose 20 people who might be promising. We brought them in for like a brutal 2-hour session where they do these various tasks. We get behavioral data, button presses, eye tracking. And then from that, we rate the subjects. We choose 15 to bring to the scanner. Due to a few dropped out, but then we actually got a full 70 session of data from 12 people doing this experiment in the scanner with eye tracking, with physio, like with the whole deal. And then we look at that data and we judge data quality from various metrics. And then we winnow it down to 6. And then we invite 6 people to engage in this very long term. So again, we're targeting 15, 20, 25, 30 sessions of data. And we're up to about 15. So it's a work in progress. I think it's a quite wacky experiment in the sense of like atypical. And I'm not sure if it's all going to quote work, but we will see. So let me pause there and see if there's any questions.
Hey, Kendrick, it's Mike.
Hi.
How you doing? Question about because there's so many different tasks and they have to recall them each time. Do you wonder if you're actually going to get some kind of frontal switching decision-making process as well, kind of looped in there? Because even if you're very practiced, you sort of feel like you're going to have to go, okay, no, I'm going to change my disposition for the particular task at this particular moment, and then what my predictive actions are.
Yeah, I think task switching is for sure going to be a big component of this data set. Are you intimate? Whoops, I went too far. Are you thinking of this as a problem or a feature?
No, not. It's just kind of an interesting question that it's a little different from naturalistic behavior in the sense that it's just something to be aware of, right? Because we typically react to what's happening in the world, although sometimes we're anticipating. But here it's going to be a sort of anticipation and code switching kind of behavior on every trial. It
Yeah, I
almost be
agree,
interesting
Albert.
to do a
Yeah.
run where they're very practiced, where you give them in some way where they have to purely react and there's no cue about what's going to happen until they see it.
I think that's pretty interesting. I think we went all in on the issue you're getting at, like non-naturalistic task stuff. So yeah, so uh, I think it would be interesting to design a whole new experiment tapping into more naturalistic tasks in the sense that you just said. But
Yeah,
yeah, hard design decisions. Like I
yeah,
met
no,
a
I
bunch of
understand that. It's philosophical more than anything. I guess also is the scan. I forget the scan parameters are such that you're scanning during the period where they're prepping for the task as well, right?
yeah, so this entire QuickTime movie represents an actual, it's just a screen recording. So it's 6 minutes
Right,
long. And yeah, we scan from the start. So this is pre-scanner. There's actually an eye tracking calibration in the scan itself. I think that's going to be super useful just to double check that everything makes sense. So there's some target saccade stuff at the beginning of each run. But meanwhile, yeah, we're scanning this whole thing. There's actually a pupil, like
right.
dark
So
light
that
pupil.
prep time before each task, where you're aware of the task, but you haven't actually had them perform it yet, could be really fascinating too, as a separate analysis almost.
Yeah, I agree. Unfortunately, we didn't the tasks start almost immediately. You get 4
Oh,
seconds.
that's what I was wondering.
Yeah. Yeah. So let me pause it. So you get 4 seconds to read it. So we allow eye movements. We don't want them to forget what button is what, obviously, otherwise it's almost useless. So you get 4 seconds to do that, and then after that, the trials essentially start immediately. But I think it will be possible to, in some sense, model out the
Right. I mean, there's
whatever
some
your
really
brain is
interesting
doing.
predictions
Yeah.
of predictive coding there that could be really quite interesting.
Right.
So,
Any
all right,
other?
very cool.
Yeah. Thanks, Mike.
Yeah, Kendrick, it's John. Sorry I was a few minutes late. So I might have missed something, but I had a similar thought to Mike, and I'm wondering, rather than, of course, going to fully natural tasks in another time is a whole other thing and has value and it's probably beyond the scope of this. But there's maybe something intermediate between what you're doing and what he's asking, or maybe he was even thinking this, is that there's a question, I guess, if of is the response to these trials independent of the timing of the task switches, so that I forget what it is right now, but the task changes every trial. That's some small number of seconds. If that number of seconds were just tripled, but it were otherwise the exact same task, or times 10, so it's a whole run of the same task, how different might the responses be, or how similar? And so then I guess the question is it practical to devote one session to just studying the timing, where you pick some maybe smaller number of tasks and you just have a very different timing from what is in the main experiment?
Good point. I haven't thought about this. Maybe we should chat more. So I should mention, I didn't know, John, you joined. So we do have a little list of like friends of VCD. We have pinged John, for example, on designing
And
and
I'm sorry that I'm
research.
never quick enough to respond until it's the middle of a talk.
Yeah, no worries. Yeah, we should chat about this. I think we're open to it, and we're, in a sense, we're not done, right? And so there's time to add some new manipulations. I will say one thing, given that this has come up now with you and Mike. There are catch trials. So we introduced, think of it like this: you get the cue, and you don't get a stimulus, or you can think of it as a gray stimulus everywhere. So there are occasionally trials where your brain is ready to do this thing, and you don't actually get any visual stimulus. So that might be useful and interesting to look into.
I have a question. Have you thought about learning during the experiment? I was thinking about the objects, the natural objects. Can you discriminate from the next? So obviously, if it's outdoors versus indoors, you have no trouble. But if it's two versions of fruit salads, you have a big trouble, right? So that would mean if you have repeated trials with the same stimuli, you could see how learning occurs to the
Yeah.
extent there's perceptual learning. Well, that's just a vague thought, but I was just looking at your design of this experiment. With your 6 subjects, you've got this great long period of time of repeating. So is it possible within that data set to find something interesting about which parts of the brain are changing, when perceptual learning occurs?
Yeah, great question. I just popped up a slide. It's not polished, of course, but we're tracking performance, behavior, MRI, blah, 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 across our data collection effort. Each number is a visit to the scanner, so we're up to
Wow.
like 15. One thing we did see is, and yeah, it's very tricky how to exactly quantify this, but I don't think it matters.
Right.
I think one thing we're noticing is dropping. So this is probably, my guess is it's learning. Like the very first session, they were like, everything was novel and like they were on
Yeah.
fire and they were working hard. And then somehow, their brains get more efficient or they learn or something. But there are these fluctuations. I will say, what accounts for this big seeming discontinuity here? Well, we introduced long-term memory and imagery right here. So they had to learn these new tasks at this point, which required a whole training, and then we brought them
Wow.
back in. So some of these subjects showed this big change. Take it with a grain of salt, of course. This is work in progress. But yeah, I think there are signatures of learning, which I didn't really want to sample, but I think it's inevitable, probably, given how strange and crazy this behavioral battery is. So yeah, short answer is it's probably in there, but certainly we haven't analyzed it or figured it all out.
Maybe you could take the average bolt strength as the denominator and then the area connected to, say, color perception or something like that as the numerator, right? And if you've got a ratio, and because you're getting better at the task, so you don't need so much blood flow, but relative to that, there might be some particular changes low buried in the data. It might be possible to find interesting examples.
Yeah, for sure. I think it's super interesting.
Going up, right.
But yeah, behavior also is showing a general upward trend across scan sessions.
I mean, this is across all of the tasks, right?
Yeah, this is not broken down by task, although these columns are, because we're monitoring long-term memory was its own thing, and like we were worried if they could actually remember all the associations. But anyway, we're keeping tabs on what's happening, and there are interesting trends, I guess is the summary.
Yeah.
And just
sorry, I'd like to suggest maybe we continue and leave, just see that you get through the
Yeah,
talk, and we can do
exactly.
the discussion at the end, if that's okay.
Right. Yeah. So as usual, I have 9 minutes to do this. So I will just give you the flavor. I'll give you the flavor of this. Oh, okay. Well, we'll see how it goes. Let me just give you the overview of this last project, I guess. They all have 3-letter acronyms. I don't know. Seems useful for our memory. So the theme here is, I guess, data. We want better data, high field data, blah, blah, blah. So PSN is a denoising technique in short. And it's something that I find interesting. So let me just give you the overview of how it works. Basically, I seem obsessed with trying to get the best possible signal. And so what data, largely speaking, can be divided into there's noise and there's signal, defined as the signal is typically what we want. We make a manipulation, we record multiple trials, we try to average as many trials as we can. So it's kind of the expected neural response to a given manipulation. Everything else, all the variability, all the residuals, let's call noise. But noise doesn't mean not interesting. There's many, and I will just fly through this. There's many kind of reasons to think that neural activity fluctuations from trial to trial might actually be useful and meaningful, or the brain might actually use these fluctuations to do something. So noise doesn't mean bad necessarily. Certain types of noise are bad. Like you don't want head motion in your fMRI data set, presumably, or unless that was actually interesting to you. So anyway, there's good noise and there's bad noise, and currently we're just lumping it all into the noise bucket. And from a statistical perspective, it would be nice if we could just figure out what is the actual signal and what is the actual noise. In some sense, this is impossible because if you could, well, that's why we get data. So you get 5 trials, 10 trials. If you knew what the expected response was, that's essentially saying you got infinite data to average out. And that's obviously impossible. But there are some ideas of what we can do to more closely approximate the signal. And that could be very useful, especially in neuroscience data. Okay, so there's a lot. Just the overview of how we approach this is the following. So the precursor to the denoising technique was this first paper called Generative Model of Signal Noise, or GSN. It's actually a really simple technique. This is a collaboration with these folks. The basic idea is as follows. When you stop to think about it, I think, can, for the most part, think of most neural response measurement data as essentially a sample of signal and noise. And then the goal is let's estimate what these underlying distributions are as a first step. There's some statistical ideas here that is actually pretty simple. It's just a summation process. And the idea is we can observe the data, which is a mixture of signal noise. That's easy. We can observe the noise, which essentially you subtract off the mean response across multiple trials. And essentially things summate, so you can estimate the underlying signal by just subtraction. That's the basic idea. I'll show you a diagram of this process. So imagine you're in multi-dimensional response space. So the axes are neurons or voxels or channels, doesn't matter. And in a given experiment, we're trying to drive in vision, let's say in a stimulus experiment, just like different spots in this high dimensional space using different stimuli. So the signal might have a certain distribution. The noise, of course, has a distribution, trial-to-trial fluctuations. And the observed data is essentially the summation of these two distributions. So if we can estimate this and we can estimate the noise, then essentially we can subtract the covariances and get a reasonable estimate of what the underlying signal is. That's the basic idea. There's a little bit of trickiness in terms of the math. So one thing we show in the paper is typically we just analyze the data and we hope there's not a lot of noise if the object of interest is the signal. But that's a naive approach because we know there's always noise. And we show one of the consequences of doing this technique is better estimates of dimensionality. So this is just an eigenspectrum. So essentially looking at the PCs of a set of data. This is real data. And we just show, for example, if you do nothing and just average across trials and look at the eigenspectrum, look at the essentially variance explained by various number of dimensions, you get a certain sense of what's going on in the data. Versus if you decompose the data in the sense I just said into signal and noise distributions, they actually give you two bits of information. You actually separately see how the signal and noise are contributing to your dimensionality. Okay, so that's the first step. We can get these distributions. The question is, as I said a second ago, I know I'm going fast, is how do we actually denoise our data? We can average across trials, and that we hope is going to, on average out some of the noise. But there's still noise left in that trial average thing. So how do we do better? Well, the insight then in this project is to essentially use a classic idea of low rank denoising or PCA-based denoising. So for probably most of you will get this. You can apply PCA to data and learn the directions in your data that are responsible for most of the variance. And we hope that we have enough signal in the data such that the first several PCs carry most of the signal. And if that's true, then we can essentially zero out the remaining dimensions and as a way to possibly denoise the data. Like basically beyond dimension 10 is probably just noise. So let's just discard the variance out there. So that's a very sensible classic idea. We're calling it naive PCA-based denoising. But the problem with that approach is the noise sources in your data set, if it's EEG or fMRI or single neurons or whatever, the noise actually might be in the first couple PCs, right? So naive PCA, in a sense, makes no distinction between what is signal and noise. Okay, so that's a problem. And so the idea then is, well, we just got done talking about GSN as a way to kind of guesstimate what the signal and noise distributions are. So let's focus the low rank process on the dimensions of the signal estimate. That's it. I mean, that's it. There are a lot of details, but that's the core insight. So the key idea is let's use this kind of low rank denoising idea, but now instead of just using the naive PCs, let's just use the PCs of the signal estimate. So very general, has nothing to do with brains or anything. And I think that's a virtue because we have applied this to many different types of neural measurement modalities, and it seems to be very robust, very versatile in that respect. I'll show you a little animation since maybe I'm talking really fast, so it looks like I have a minute. I'll give you a geometric intuition of
No,
how this
it's
all works.
okay if you go over time. Don't
Okay,
worry.
well,
Don't.
okay, well, I'll slightly slow down. All right, I just want to give you a schematic of how this actually works. Hopefully it will make sense. Or I think geometrically, so I designed this figure to give a geometric intuition. So same example. So we're looking at this fake three-dimensional space where the signal and noise distributions have different shapes. Fine. So let's imagine the following situation. This is the true signal without any noise. And then unfortunately, when we measure each of these conditions or stimuli, whatever they are, we are corrupted by noise. So here are a set of measurements where I've taken, shown this stimulus and added some simulated noise on top of that. So we get this kind of cloud of data points that don't live on this nice plane as I drew it here. So the black dots are essentially the data distribution. Okay, so how do we recover the underlying signal, or at least better approximate it? Well, notice the signal comes from this plane, right? I kind of drew these points to live on this plane, so I'm just going to show you what that plane looks like. We can take the data points and project it down onto this subspace. So this 2D plane that lives in this 3D space, we can simply project those data points down, and that gives these blue dots. And that's nice because the blue dots are closer to the underlying signal, the red dots, than the original black dots. And we can show that with a little rotation. So let's rotate this space such that we're looking at the plane from dead on, so it has no width anymore. So we just rotated the space a little just to show you that, yeah, the blue dots live on the plane, great, and they're closer to the red dots than the original data points are. Okay, that's the ideal case where we know the signal subspace and that's it. We just project it down. Of course, there's some tricky issues like how many dimensions is the signal subspace? But that's the core idea. And I'll just show you, for contrast, what happens if you do the naive approach. So if you just take the data itself, so the black dots, so it's the same black dots here, same underlying signal here. But if you just do PCA of the observed data, so not using this whole GSN intermediary, if you just do standard low rank denoising, uh, the data, I'm calling it the data subspace. So it's not the underlying signals subspace, it's just the observed data subspace. If you do that and do your low rank reconstruction and do this rotation just so we can see, same data, but we get a different denoised version of the data. So those are the blue dots. These blue dots are closer to the red dots, but they're not as close as the optimal thing to do, which is use the actual signal subspace. So hopefully that gives you a sense of what's happening. And so the premise here is, yeah, if only we knew the signal subspace, then it seems obvious that we can better denoise our data than doing the naive approach. That's it. There are a lot of details we're working on shoring this up, being rigorous. We can run ground truth simulations. Jacob has done a lot of work on this. We're using a toy example here of what the underlying, again, nothing to do with brains, so we can use this fake case of you have neurons responding in this particular fashion. We can, generate data. We can show what it looks like if you just trial average. We can show what it looks like if you use a naive low rank reconstruction approach, which retains a lot of the noise, and then we can contrast it to what happens if you try to estimate the signal subspace and use that as a target. It obviously looks better. Another thing we're doing, which is really critical, is like this is just proof by eyeball, but not everything you see is necessarily trustable. There's this issue of bias. Like you don't want to do a denoising method that somehow is introducing biases to the data set that you produce. And so there's a whole series of bias variance simulations. I actually wrote a whole paper on this just to kind of lay this out there because I think it's often ignored, especially with the advent of complicated AI and machine learning techniques. They are effectively introducing biases that can be very risky. So anyway, there's a whole series of bias variance simulations. I'm not going to explain this other than we are taking this super seriously and carefully tracking the biases that various flavors of these types of methods can introduce. So I think that's important. And as I said, we have gotten a lot of success thus far applying this to vastly different data sets. Hopefully it's clear that, again, there's nothing in this technique that makes it specific to the types of noise in fMRI versus EEG versus whatever, which I think is a major strength. And so it looks like a very versatile tool that possibly can be used off the shelf on any given data set that you have. Okay, so just to wrap up, kind of started off with introduction of how we think about trying to understand the visual system. And the idea of collecting these very large data sets, neural data sets in a small set of individuals. I briefly reviewed what we did for NSD, where the focus was on stimulus diversity. And then I went through VCD very briefly as this kind of ambitious data set that's trying to cross stimuli with tasks in a kind of classic controlled trial-oriented paradigm. And lastly, I just got done with a very quick overview of this computational neuroscience method that we're developing to better estimate signal in a given data set. That's it. This is the lab website. There's a bunch of stuff there, including code, data, and other things. Thanks.
Really great talk, and I want to invite everybody to unmute yourself. Let's give Kendrick a big applaud for a really, really great talk. And if you're up to taking questions, I'm sure there will be several, and I'm happy to I'm happy to start. I have two.
Sure.
So first of all, I wanted one thing that is related to the VCD, if I'm not mistaken, with the letters project, following Mike's idea and Adam's, and maybe what Peter was thinking about saying is about and about the tasks is, I think if you're still collecting, it may be interesting to also just include naturalistic vision, not telling the participants like they can read the instructions, but they're not, they don't really have to follow any, it's a free few observation scans so that you can basically, and maybe even repeat that twice, so you can like have some sort of baseline, although it may not be the perfect baseline because they already have this learning, but still. And I also thought maybe it would be relevant to examine their eye tracking outside the scanner for these images. So you can then, that's much higher precision, resolution of how they scan the images. And then maybe you can use that to model some aspects of the brain response that you may not be able to do based on the probably lower signal precision of the eye tracking within the scanner. And I also wanted to ask with respect to this project and to the PSN, you are basically in the VCD, you're looking at two dimensions like task and stimulus. And from each point of view, in a way, you can think about the noise as being something else. Now, maybe you can think about the two-dimensional noise because if you're thinking about the stimuli, it's, the task would be potentially noise. So I'm thinking, would you expect in your last project to have, like, would you combine the noises? Would you examine, like, I don't, maybe I'm not fully
No, I think, yeah, I think
polishing
I get.
my,
No,
yeah.
no, yeah. Thanks for the ideas. Just the first couple things you mentioned, yeah, super interesting regarding the eye tracking. I'm a little torn with the idea of sticking him in there without any overt task. In some sense, I railed against this in the middle of review. Like, what is that? What are we even doing when that happens? I mean, you're allowing the subjects to choose their own task, but you don't even know what that is, right? But I can definitely see the value. Like that kind of also makes sense, or it might be interesting to sample. So yeah, I guess we'll think more about that. Just to clarify, in VCD, they are asked very strongly to fixate the whole time, even for the natural scenes. So in the
Oh,
scanner,
okay.
they, in theory, are not moving their eyes much at all.
I see.
Um,
Okay, okay.
I will say that outside the scanner, we are somewhat, although we're a little new to this kind of area of thinking about vision and stuff, but we did get other participants to come in and look at these exact scenes, doing these exact tasks, or some of them at least, and allowing them to move their eyes. So sample what humans do without the constraint of keeping your eyes steady while doing these same tasks. So we have some of that data. Not entirely sure what exactly to do with it, but we were a little playing around with it. And then your last bit about the noise bit. Yeah, I think there's many different ways that one can apply these ideas. I think the most straightforward way here is to essentially be at the finest level. So like for every different task type or crossing, like
Mm,
think of noise as the fluctuations while subjects are engaged in that. Certainly there may be differences in signal and noise across every manipulation. So uh, and yeah, it gets interesting and complicated, right? Like noise is not necessarily bad. Noise could be interesting from a cognitive point of view. Like we know subjects aren't necessarily doing the exact same thing every trial, duh, right? Because there are behavioral fluctuations. So I think there's a lot that can be done mining that point of view. Like, can we track essentially the fluctuations in the trial-to-trial neural data and link that to behavior, which many people have done. Um, Anyway, and there's also like, manifold points of view of like, is the noise actually changing across tasks? And is that, in what brain regions? And like, does that support behavior? There's a lot of like, yeah, that type of thing
yeah.
that can be done here.
Yeah, it's a very rich, I mean, rich data sets, not only one of them. They're all, yeah, very rich and interesting.
Mm.
Thank you. There are two, so Michael, I'm not sure if he's still here. So there are a few comments in the chat, but Mike
Oh,
Tar wrote, I mean, maybe you
yeah,
can,
I can take a look.
okay,
Um,
okay.
let's see. Um, Looking at Mike's comment, behavior is driven by the stimulus context. Yeah, in the current VCD design, that's not really possible because as a matter of design, every stimulus participates in almost every task context. So it's not obvious if you just see the stimulus what task to do, which is a major problem, of course, because the subjects might forget what task they're doing in the middle of the experiment. I think for the most part, this things have been going well in the sense that subjects task switch rapidly. I mean, this was kind of one of the points of doing the whole psychophysical session. Is it actually possible for a reasonably smart human to remember all these tasks and do it at a rapid pace? So I think we're pretty confident subjects are doing well. Occasionally, subjects say, if you've ever been an fMRI subject, fMRI is hard, let alone doing this experiment at the same time. Subjects occasionally tell us, like, whoops, in one block, in this one run, I actually forgot what task I was doing. So they did admit that, but these are pretty rare. Yeah, and again, I just want to emphasize, like, it was not, oh shoot, I just stopped it. The subject selection process, we put a lot of thought into it, like, and we took it because we were worried. I don't know why I can't share. We were worried, you know, if you stick a normal human without a lot of training for this, they're just going to flail in the scanner. So we did do a lot of screening and training. There was a whole like hour-long class essentially. So the RAs I have on this, they made a whole presentation. Like, look, here are the stimuli. You have to understand what we're doing. You know, you have to understand what motion direction is. You have to understand what fixation means. You have to understand what MRI is. You can't move your head, like all this stuff. So there was an hour-long video that they produced essentially to try to instruct these subjects and make them suitable for this experiment.
Kendrick,
Yes.
I have a couple of questions and I can't find the little hands, you know, the one that you raised. I don't know where it is here in this interface. Anyhow, so one question I had for the VCD data is the way they respond, just in terms of like what they press and how many buttons and options, is that the same across the different tasks?
No. Yeah. So if you wanted to perfectly balance all that, essentially one of the themes in design is they said it's nice in general, of course, to balance various things, but in the end, it's impossible given the many dimension. So I wonder if I can pull up the figure or not. So if you look at a histogram of like what is the correct response, some tasks only have two alternatives. Some tasks has four alternatives. There's one that has three button presses. And then we are trying to, in general, achieve balance. So you equally press ones and twos, but sometimes they're slightly imbalanced. So yeah, that's the short answer to your question.
Okay. And the other question I had, it's again in relation to that data set. So, I mean, have you considered and maybe excluded for whatever reason the possibility of having some of the stimuli that were auditory? Because I would have thought that if you're trying to look at task invariant, I mean, task-specific activity, maybe you would want to change the stimuli even more in a more extreme fashion, like, you know, doing the same. I understand that then again, you have the problem of what is the same task with a visual stimulus and auditory.
Yeah, sorry, you just paused, but I think I got the gist of the question. Yeah, no, so, oh, sorry, your audio just came back. But yeah, no, this is all visual stimuli only.
Okay.
I guess, sure, that's the next data set. Let's do a multi-sensory one.
Okay. Thanks.
Great. So I really want to thank you a lot, Kendrick, for joining us and for giving such an interesting talk. And thanks for sharing your science with us. And I hope I want to wish everybody a best rest of the week. And
Yeah, great. Yeah, thank you for all the comments. And I actually want to watch this recording just so I can jot down the good ideas that came up.
yeah, definitely. You will definitely get a copy. The issue is if the others will. Yeah, great. Thank you
Okay,
so much.
thanks for your
And
time. Okay,
Thank you,
bye.
guys.
thank
Okay,
you.
congrats.
Bye-bye.
Bye.